this is Ravindra Babu. Welcome back to my channel Silver 90s by Unit. In this part of videos of molecular basis of inheritance, today we are going to discuss the next concept under this, that is RNA world or dominance of RNA in the primitive life existed on the earth. In the last video, we are discussing about the 4 characteristic features to which a genetic material must have. To those characteristic features, we searched in both DNA and RNA. Finally, DNA won the contest by having 4 characteristic features of a genetic material. But RNA fulfilled only one criteria of genetic material. In the remaining 3 criteria, it was failed. So that finally we concluded that DNA is a predominant genetic material over RNA. Now again, 2 questions arise in our minds regarding DNA and RNA. Those were, the first one is, what is the first genetic material evolved on the earth? Among this DNA and RNA, which has evolved first on the earth? That is the first question. And the second question is, what are the evidences to suggest essential life processes such as metabolism, translation and splicing have evolved around the RNA? The answers for these 2 questions has given by a hypothesis, that is RNA world hypothesis. RNA world hypothesis. This hypothesis was proposed by Alexander Rich in the year 1962. In the year 1962. According to this theory, RNA world is an hypothetical stage in the evolutionary history of life on the earth. And this theory says that self-replicating RNA molecules proliferated before the evolution of DNA and RNA. So that this theory considers that the RNA is a first evolved genetic material. This hypothesis explains the dominance of RNA in the primitive life of Earth in the following way. It considers that the primitive Earth consists of primary elements such as carbon, hydrogen, oxygen, nitrogen and phosphorus. These primary elements of a primary Earth gradually in the process of evolution they could able to form 3 components of nucleotides. You know very well the 3 components of nucleotides. One is nitrogen base, phosphate groups and pentose sugars. The first formed nitrogen bases are adenine, guanine, cytosine and uracil. So later in the process of evolution, these 3 components could able to bind with each other to form ribonucleotides. Say consider these are the ribonucleotides. These ribonucleotides are free floating. These free floating ribonucleotides was considered to be as a primordial soup. What we call it? Primordial soup. And later in the process of evolution, these ribonucleotides gradually they bind one after the other to form a polyribonucleotide chains or RNA strands like this. These are the polyribonucleotide strands chains or RNA strands having a polymer of nucleotides like this. And later these polyribonucleotides whichever formed here, they gets encapsulated like this. This encapsulated polyribonucleotide strands is considered to be as a first form of life. First form of life. Nothing but a primitive life. Primitive life. The RNA strands formed in primitive life in the process of evolution underwent some conformational changes and foldings. These changes made these RNA strands to show characteristic features like replication and catalysis. The RNA strands whichever being formed here shows different replication outputs. Nothing but some RNA strands, some RNA molecules can undergo replication efficiently and can increase their number through this replication, but some of the RNA molecules, they may not have that much capacity to make their own copies. Such type of RNA molecules may not survive, they will be washed out at this stage of evolution. This is what generally we say natural selection or survival of the fittest at molecular level. To this we can call it as molecular competition. Additional to this replication capacity, here the RNA strands also got the catalytic properties through mutations. So mutations made these RNA molecules to persistent and expansion of their number in this molecular competition. This molecular competition among these RNA molecules favored the emergence of a coordination and cooperation among the different RNA molecules, which opened their way for the formation of a protocell. This is a protocell where RNA can show a replication property. The same RNA molecule can also involve in the synthesis of proteins. Suppose these are the amino acids. Here RNA molecule can bind these amino acids together to form a protein molecule. Nothing but first RNA molecules got the catalytic properties. By these catalytic properties, RNA synthesize the proteins. So proteins gets converted into enzymes. These enzymes later became predominant biocatalysts over RNA molecules because they consist abundance and diversified monomeric units when compared to RNA molecules. Let's say enzymes. Enzymes are made up of amino acids. These amino acids are actually abundant in the nature, and these enzymes are made up of 20 different types of amino acids. Nothing but the enzymes are having abundant and diversified monomeric units as their constituents. If we come to RNA molecules, these are made up of ribonucleotides, which are less abundant in the nature, and that too there are only 4 types of nitrogen bases or 4 types of nucleotides are involving in making of a RNA molecule. In earlier life, before the evolution of proteins, all the biocatalysts are ribozymes. So therefore the presence of ribosomes is ancestral characteristic features in the living organisms. Suppose for example, that is a 23 is a rRNA which involves in the formation of peptide bonds, which acts as a peptidyl transferase, involves in the formation of peptide bonds between the amino acids during the translation process. In earlier days, RNA being acting as a biocatalyst, it combined the amino acids together to form a protein molecule. This reaction in the presence of RNA, these proteins gets converted into enzymes. These enzymes involves in a biochemical reactions. Some of the biochemical reactions of a living organism is said to be as metabolism. Nothing but in the earlier days, the metabolisms are evolved around the RNA molecule. By this, we made clear the first question, that is what is a first genetic material, and in the second question we made clear that metabolisms are evolved around the RNA molecule. Along with the metabolisms, the splicing mechanism also evolved around the RNA molecule. In order to understand this, we need to go for a transcription and post-transcriptional modifications. Say, this is a DNA molecule. This DNA molecule consists both coding regions, exons, and non-coding regions, introns, alternately. In exons, introns, exons, introns, exons, introns, like this. Exons are nothing but the coding regions, the portion of DNA which involves in the expression of a character, that is what a coding region, exon. And introns are non-coding regions, non-coding regions, of DNA or RNA which doesn't code for any character, that is, and this DNA undergoes a transcription. During this transcription it forms a hnRNA molecule. This hnRNA means heterogeneous nuclear RNA, nothing but the hnRNA got the genetic information as such from DNA through the process called transcription. So that here also you can see exons, introns, exons, introns, exons, introns, exons, introns, like that. This hnRNA molecule, it undergoes post-transcriptional modifications. During this post-transcriptional modifications, it loses introns by a mechanism called splicing. The splicing mechanism is carried out along with capping and tailing, where tailing process gives a tailing process provides a 7-methyl guanosine as a cap region at 5' end, and a tailing process provides a poly A tail at 3' end for a mRNA molecule. This is mRNA molecule which consists only the coding regions, exons. If you observe one thing here, both the DNA and RNA are having introns and exons, or both DNA and RNA are showing a split gene character. Split genes means the coding regions are splitted by non-coding regions. That is what we call a split gene character. Both DNA and RNA are showing this split gene character, but in the process of evolution, RNA got the capacity to get rid of these introns or non-coding regions by a process called splicing, but DNA it retained the non-coding regions as such. So the process of splicing, which is evolved around the RNA, represents the dominance of RNA world, and presence of these introns in the nucleic acids represents the reminiscent of antiquity. And third essential life process, that is translation, also evolved around the RNA molecule. If you carefully observe the translation process, the mRNA which was formed after a transcription, it undergoes a translation. The translation process requires all the 3 types of RNA molecules. The mRNA itself is undergoing a translation, and along with this mRNA, the tRNA molecules and rRNA molecules also involves in the process of translation, so that the 3 combinedly work together to form a protein or polypeptide chain. There is no involvement of DNA here. mRNA, it translates the genetic information from nucleic acids to amino acid sequences, and the tRNA molecule acts as a adapter molecule which brings the amino acids from amino acid pool to the site of protein synthesis, that are ribosomes, and rRNA molecules which are the structural components of ribosomes. The ribosomes are playing a key role during the process of translation. This RNA world hypothesis also said that the ancient genetic material RNA in the process of evolution got changed into DNA by undergoing some structural and chemical changes. These structural and chemical changes are, one is by becoming a double stranded structure first, becoming a double stranded structure. This is a structural change came in RNA, okay, and the second one is losing of oxygen at a 2' position of ribose sugar. so that ribose sugar became deoxyribose sugar. And third one is getting thiamine at the place of uracil, okay, which made the genetic material to involve in repair mechanisms. And fourth one is developing self-repair mechanisms. Actually, the ancient genetic material RNA does not have these characteristic features. By getting these characteristic features, the RNA became a DNA in the process of evolution, so that this DNA is acting as a perfect stable genetic material today. Thanks for watching this video. Please like, share and subscribe my channel.